All right, guys. So this is a walkthrough for the gene mapping activity. It's from pages 41 to 45 in your notes packet. I'm not going to walk you through all of it, but I will walk you through a little bit of it to get you started, and hopefully it will help you move right along. All right, so we're going to start at the beginning here. You can read all of this stuff up here. Really, the most important part is this last sentence right here, especially for answering the first question. So we have cross perform between these two parents. Number one says predict the genotypes of the F1 progeny. This is actually pretty easy because if you notice here and here, we have true breeding parents. Okay, so with true breeding, when that happens, the genotypes of the F1 progeny are always going to be the same. So here, our genotypes are going to be heterozygous. Okay, now question 2 looks very confusing. Let me just get this situated. Question 2 looks very confusing, but I don't want it to confuse you too much. Essentially, what it's asking for is it wants to know what the products of meiosis would be based on the F1 progeny. And essentially what's happening here is that you would kind of do this like you would do the dihybrid crosses. So if you had two F1 progeny that you were crossing, what would those genotypes be that you would use on the dihybrid cross? So this is when we use our foil like this, right? Like that. So we have, first one was done for us. So we have dominant R and T. That's the first one right here. The next one we will have is a dominant R and a recessive T. And you can just kind of, do that number right there. Okay. Then what we would have after our dominant R and recessive T is we're going to have dominant T, recessive R. And then for our last one, we're going to have both recessive, like that. Okay. Now down here, this is essentially going to be kind of a dihybrid cross. The thing is, though, is that it says an F1 individual. So we know that that is this right here. And then it says it's crossed with somebody that is homozygous recessive. So you can, or really what's supposed to happen is it's supposed to be a dihybrid cross. But because our other parent is homozygous recessive, we're going to use these same genotypes that are here for our test cross. The nice part is here, we only have one option really, and that's this, right? There are no other options given. So instead of having to do a full-blown 4x4, it'll kind of be like this, right? Where we have our genotypes from up here on the top. So we have TR, TR, like that. And over here on the side, we just have that right there. The ratio of these is going to be the same because you could do a full-blown dihybrid cross, the 4x4, but you would end up with the exact same stuff, and this is just a little bit easier here, right? So now that this is set up, I'm going to let you do that, and you need to basically figure out what the ratios are. So use fractions for this to give your answers, and be specific about the phenotype, right? All right, next page. So here, this is page 42. You're going to read this part up here, right? And then number 4 says, refer back to the ratio of phenotypes of the offspring generated from the test cross in question 3. How would these ratios change if the genes for seed texture and plant height were linked? So law of segregation says that things are independently assorted. Okay, So essentially what would happen here is the percentage of parental phenotypes would increase. And the reason why is because the linked genes would be inherited as a unit. If you need time to write that down, go ahead and pause the video. All right, so the next part here is that now they're talking about, okay, what if we have two new traits? These traits are the shape of the pollen grains and the number of leaves, and determine whether these traits assort independently or are linked. They're going to cross them. And that's kind of what's happening on the lower half of this page right here. So let's look at it. We've got our P generation, our F1, and then we do a test cross right here. And we've got our genotypes there. And they already gave us the observed numbers for this cross. And then our total is 2,700 total, okay? So our expected value here would be if all of these categories had equal numbers of individuals. So what you would do is you would take your 2,700 and you would divide by 4, and the total there would be 675 for all of them. Okay. All right. Let's go ahead and flip that page. So this is page 43. These are some questions here for you to answer. I am going to go over number 6, and I'm also going to go over number 7. Okay. So number 6, here's what it says. It says, using the data chart on the previous page, label the offspring as parental phenotypes or recombinant phenotypes. So here's what that means. Okay. We have our parental phenotypes here. Let's see if I can get it up there. There we go. All right, so if you look here, here are our parents for this test cross. And we have one that has 2 leaves and the short pollen, okay? And then our other parent has 1 leaf and the long pollen grains. So down here, comparing to our offspring, this one also has 2 leaves and a short pollen grain. So this right here is a parental phenotype. And then this one right here matches up with this guy. So this is also a parental phenotype. So it matches up with whatever the phenotype or the physical traits of the parent are. That means that these 2 guys here in the center, these are recombinant, which means that because of genetic diversity, because of crossing over, that the genes have recombined, and you end up with traits that are different than the parental phenotype. So that's number 6. Now, number 7 said, using the observed data, calculate the recombinant or recombination frequency, which is the number of recombinants divided by the total offspring. So here's the keyword for this one. The keyword for this one is using the observed data. Okay, you gotta read really, really carefully on these questions. So if you look back at your observed data for recombinant, so here are recombinant phenotypes right here, and then our observed are 350 and 334, and that number is 684, and we're going to divide that by the total offspring, which was 2,700, and that gives us 0.25. You can either multiply by 100 or you can just move your decimal. This is 25%. Now, one of the things that it talks about when it comes to gene mapping, if you remember from the notes on page, it is on page 40, that 50% recombination rate is, that means that the genes are really, really far apart on the same chromosome, or they're on 2 different chromosomes. So this is less than 50%, and that will help you answer number 8, but you got to answer number 5 first, okay? All right, let's keep it moving. So now we'll come to the fun part, which is where we're going to make a linkage map. So based on data, let me see if I can get this to focus. There we go. So based on the data, we're going to create a linkage map. This first one is really helpful. So essentially what you're going to do here, it says start by finding the genes that are furthest apart. In this case, using this data right here, C and E are 40%, which means 40 map units. So 1% recombination frequency is equal to 1 map unit. Okay. So what's going to happen here is what they did was on this gene, they went ahead and put these about 40% apart, or 40 map units apart. And that was that one. Now, what you're going to do for the other ones are, you need to look and see what they're trying to connect here. So the next one is that we have C and D. So the distance between C and D, the recombination frequency is 35%. So if you notice down here, what they did was they put D very close to E, because the distance from C to D is 35 map units. So that would be that one. Then we have B and E, 25%. So we'll put B over here because we have already established where E is. That's 25. And then our last one is B through D. Okay, I'm going to show you how to do it down here. Let me get my paper settled and situated. Come on. There we go. All right. So this one right here, so it says construct a linkage map on the chromosome for each of the data sets given. So we have one set of data here with our genes, and we're going to start by looking at the one that is furthest apart. So that is our highest percentage, and that's M to J, and it's about 42%. So I'm going to put them pretty far apart. Okay, roughly. So this would be M, and this would be J. And it says that they are 42%, which means from here to here, they are 42 map units. Okay. Okay. Then after that, where our next highest frequency is M to P. So M is right here, and this is 21%, which means that it's going to be kind of about halfway in between these guys right here. So I would put it right about here, and we will label that as P, because the distance from M to P is 21 map units. Okay. After that, we have R to J, and it is 16%. That means that R and J's right here, 16%, is sort of close to 21. If you look at your last one here, we've got P and R. We already have P, and when we put R here in a minute, these are only 5%. So that means that our R is going to be pretty close to our P, right? And from P to R is 5 map units, and from R to J, it is 16 map units. All of these work very, very similarly. So do your best on the other ones and see what you can do. And if you need help, please ask for it.